Hey everybody, Troy here. Today we're going to be talking about myostatin. Is it the way to get uncontrollable muscle gain? Well, let's look into it. What does myostatin do? Well, myostatin is a regulator of muscle mass. Specifically, it is a negative regulator of muscle mass, meaning as we have more myostatin, there's decreased muscle mass. So this is usually caused by increased body growth. This is seen as in obese people. They have more myostatin. In bodybuilders, they have more myostatin. So as your muscle size, or typically body mass in general, increases, the more myostatin is expressed to decrease your ability to grow muscle. Second would be activin A. Activin A is another regulator of muscle mass. It's H released, and it's also responsible for neuroprotection. Third would be our activin type 2B receptor. So this is a receptor that both myostatin and activin A bind to transcribe their muscle breakdown effects. Fourth would be follistatin. Follistatin is an endogenous protein which binds onto both myostatin and activin A to completely stop their effects. So this is why we see increased muscle mass with a follistatin overexpression. And this kind of brings me into the next slide where we're looking at a mouse. This mouse has a regular amount of myostatin, and as you can see, it has a regular amount of muscle mass. In the middle, we have an activin type 2B receptor knockout. So obviously this mouse is much more muscular. But why is the mouse on the far right more muscular? That mouse has a follistatin overexpression, after all. But the receptor is knocked out, right? Well, the receptor has many other effects than just controlling muscle mass, and we're going to explore that later. Next, we'll look at the bully whippet, which is a homozygous knockout for myostatin, very muscular, very lean. The next would be the Belgian blue, which is a myostatin knockout. So it's very big, very muscular. Next, let's move on to the myostatin signaling pathway. So myostatin is first secreted as a long propeptide form. So it consists of the propeptide and the active myostatin form. So myostatin is kind of tucked away. It's unable to exert its effects on the activin type 2B receptor. It isn't until two of these propeptides are cut in half, then myostatin binds to itself, creating an active dimer. After this active dimer is released, it binds to the activin type 2B receptor, phosphorylating SMAD2 and SMAD3 proteins, which then cause protein breakdown. Next, let's look at what could possibly inhibit myostatin. First, we have the classic myostatin antibody. The antibody binds to myostatin, and then it is excreted. It's unable to bind to activin type 2B receptor. Next, we have follistatin and modified follistatin. So follistatin binds both, as you remember, binds both activin A and myostatin. So follistatin injected is able to sequester the myostatin and activin A, increasing muscle mass. So the problem with regular follistatin is that it only has a half-life of 90 minutes. So everybody buying follistatin from peptide websites and spending hundreds of dollars every day, and they're seeing no results, it's because follistatin has such a short half-life. We need a longer acting follistatin to see any effects. So that's why modified follistatin variants have been created, ones that evade metabolism, have a longer half-life, and then are going to obviously cause more muscle mass in the long term. Now, another thing that has also been used is a decoy receptor. The activin type 2B receptor is used as a decoy. They inject a receptor decoy into the body, and this binds everything that binds to the activin type 2B receptor. So now that we know what inhibits it, why don't we look what happens rather than increase in muscle mass downstream. So as you can see in the diagram, when myostatin is knocked out, there's a downstream cascade that leads to an increase in irisin. Irisin causes white fat to turn into brown fat. There's a mitochondrial portion, mitochondrial biogenesis from white fat into brown fat, which increases the energy expenditure. So myostatin not only increases our energy expenditure from fat mass, but it also increases our leptin sensitivity. So this means that we have more satiating signals than the average person. So we can have decreased fat, but the same amount of appetite and energy expenditure. So the energy expenditure is kind of shown by this graph on the top right. The myostatin wild type, they have a regular energy expenditure, but the myostatin knockout have way higher energy expenditure. And then on the bottom right, myostatin knockout mice have way better insulin sensitivity, glucose tolerance, fasting glucose, and postprandial glucose tolerance. Moving on, we want to kind of compare what happens when we inhibit myostatin prenatally or postnatally. So prenatally, there's an increase in muscle fiber count and cross-sectional area, but postnatally, there's not as much of an effect. From what we found is that there's an increase in hypertrophy, cross-sectional area, but there isn't a concomitant increase in strength. Now, why does this happen? We're going to kind of look into the human models. We have muscular dystrophy. So this is where the dystrophin protein is missing on the outer cell membrane. Now, in the human model, we've used myostatin antibodies, but these myostatin antibodies haven't provided any functional outcome for people that are suffering from Duchenne muscular dystrophy. So why is this? Well, we got to compare, like we've been looking at mouse model, cow, dog, whatever, but what happens in us as a human, it's completely not different, but it's just not the same. So when you inhibit myostatin in a human model, we got to consider that there's other controllers of muscle mass. So I know I spoke about activin A at the beginning of the presentation. And activin A, what's been found is that it's a more prominent muscle mass regulator than myostatin in primates and humans. That mice have way more myostatin than humans. So obviously, if you decrease or knock out their myostatin, they're going to have a way more pronounced effect than a human. So that's why in these studies with Duchenne muscular dystrophy, and even giving healthy subjects myostatin inhibitors, there's not a huge increase in muscle mass that you'd see. They didn't double in size. That's where follistatin variants kind of play their part. They bind to activin A, and activin A seems to be more of a prominent regulator in humans than myostatin. Now, there could be some downstream effects we wouldn't want. That activin A is responsible for neurogenesis and follicle stimulating hormone release. All this myostatin, activin A talk seems a little bit too good to be true, doesn't it? Now, there's a lot of concerns with negative outcomes of myostatin inhibition or activin A inhibition, and a lot of these kind of come from uncontrolled heart growth. We're going to have way more heart growth because we're taking the brakes off the system. You got to think of myostatin as the brakes of the system. It's slowing down muscle growth. So we're going to have uncontrolled heart growth. Well, they've looked at myostatin deficient mice and they haven't found that there's any heart morphology issues. So I wouldn't say this is a huge concern, but I would say it's something to keep tabs of. Now, the second one would be that there is issues with tendons. So what they found is that myostatin deficient mice, they have tendon brittleness, that their tendons are not as strong, that clearly there's an issue with their tendon function. Third would be that there's a disproportionate increase in muscle mass, but there's no strength increase. That there's just more muscle, but no strength. But for most people, that's not a huge concern. Now let's zoom out. We're going to look at myostatin from the big picture. So myostatin is kind of just one player. That there's the tweak receptor, which binds TNF alpha. We have the IGF-1 receptor. That's a positive regulator of muscle mass, which binds IGF-1. Insulin is also an agonist of the IGF-1 receptor. We have G protein coupled receptors, which have downstream effects which can cause muscle gain. And then we have our glucocorticoid receptors that are also responsible for muscle protein breakdown. So myostatin just plays one, in humans, not as significant role in regulating muscle mass. So we got to think of this as a sum of inputs and outputs. So myostatin is the brakes and IGF-1 is the gas. If you're going to cut the brakes, that's great, but if there's no gas, if there's no exercising, if you're not eating enough food, if you're not sleeping enough, there could be other systems in place that are keeping you back from muscle mass. So this is why we have to take a look at the big picture. It's not just myostatin that regulates muscle mass. And this kind of proof of concept has been shown in a mouse study where mice have an IGF-1 overexpression and a myostatin knockout. And what they found is that mice with a myostatin knockout and an IGF-1 overexpression garnered way more size than a myostatin, just a myostatin knockout mice, or a heterozygous knockout mice and IGF-1 overexpression, or just a wild type mouse. And this is shown as in the diagram on the bottom right. So the mouse on the far right has both a myostatin knockout and an IGF-1 overexpression, and it's far more muscular. They found that the muscle fiber types are changing from type 1 to type 2. So then there's going to be an increase in glycolytic demand from those muscle fibers that they are more prone to hypertrophy than a type 1 muscle fiber. So that's why they're bigger. This is why most bodybuilders use growth hormone, IGF-1, insulin. They're trying to push on the gas of the system. They're trying to gain more muscle, but they also could be cutting off the brakes by using a modified follistatin that lasts longer than 90 minutes. They could be cutting off the brakes by glucocorticoid receptor antagonism. There's lots of ways that bodybuilders can kind of promote muscle mass growth. Now, in conclusion, myostatin is not as prominent of a regulator of muscle mass in humans as it is in mice. So that's why in clinical studies, there hasn't been a significant positive outcome of myostatin inhibition. And that activin type 2B receptor antagonism, just taking out the receptor altogether, actually has caused uncontrollable bleeding, and that's a huge issue. And then third, that we have to consider zooming out, what are our inputs in the system and what are our outputs? Do you have insufficient training, insufficient nutrition, insufficient sleep, stress, etc.? That myostatin is not the only king, and we have to consider other aspects. One thing I didn't talk about in one of the slides was creatine. But creatine has been shown to decrease myostatin, but I want to discuss all of its effects, why it's important in another video. I want you guys to hit the like button, subscribe, comment for the algorithm, tell me what you want to hear. See you in the next one.